the clinical laboratory, the basic process starts with the specimen processing. Here is the endotracheal sample that has been taken by endotracheal aspiration. First, the plate were marked with the lab ID number, which is used to trace the sample until reporting. Moreover, the date and time of processing is noted, which is used to analyze the colony growth rate. After marking, the loop is heated. A loop full of sample is taken. Another inoculum is inoculated by spreading the sample in one side of the plate. The primary streak should be done using the whole mother inoculum. After that, the plate is slightly rotated for each secondary and tertiary streaking, and the tile should be done in a zigzag way. Between each streaking, intermediate heating of loop is essential to get an isolated colony. And also, the streaking should not be touched in the edges to avoid the cross-contamination. For different types of sample, different types of plates were used to isolate the significant pathogens easily. Here, the blood agar, McConkey, and high chrome media were used to isolate the infectious bacteria. Moreover, for fungus, sabrodextrose agar were used. Then the slide is marked with the lab ID number for the primary diagnosing process, that is gram staining. A half-loop sample is spreaded in the marked area of the slide. The smear neither over thick nor to be over thin. Then the slide is heat fixed for further process.
The primary and most important diagnosis method is gram stain, which is used to start primary antibiotics for the patient. For gram stain, the reagent used were crystal violet iodine decolorizer that contains ethanol and acetone in 1:1 ratio, and finally the counterstain safranin. The prepared heat fixed smear was taken. The primary stain crystal violet is poured covering the smear. The positive charge crystal violet reacts only with the teichoic acid, which is present in the multiple layer peptidoglycan in gram positive organism. The slide is water washed. The second process involves pouring the iodine in the slide where it's formed crystal violet iodine complex. The crystal violet iodine complex gets deeply trapped into the multilayer of peptidoglycan in the cell wall of gram positive bacteria. Whereas in gram negative, the crystal violet iodine complex are easily washed off due to monolayer nature of peptidoglycan. In the next step, the decolorizer added to the slides in drops until the movement of stain is observed. After that should be an immediate water wash of the slide. The decolorizer shrink the gram positive cell wall, while the gram negative organism stains were removed. The counterstain safranin is added to the slide, results in staining of gram negative organism. Finally, the slide is water washed and kept for air dry. Observation of smear involves identifying the organism and also mainly to see whether the sample is collected properly.
Here is the Vitek to compact system, which is used to identify the pathogens at species level. Moreover, gives the minimum inhibitory concentration level of antibiotics according to the CLSI guidelines. Minimum inhibitory concentration is nothing but
minimum level of antibiotics is used to kill the particular pathogen. The system works under the principle of calorimetry and turbidometry.
The materials used for processing Vitek to compact involves density checker, saline, identification code, antimicrobial susceptibility test code, and isolated pathogens from patient specimen. First, 3 ml of saline is filled in the empty polystyrene tubes. Then the tubes were marked with the lab ID of the specimen from which the organism is isolated. First tube is for identification and the second tube is for antimicrobial susceptibility. The first tube is inserted in the density checker and bring it to 0 density. After that, the isolated colonies from the plates were taken using inoculation needle and mixed in the first tube without colony precipitation. Then the colony is mixed using vortex. Then the density is checked using McFarland standard. For bacteria, the density standard is 0.50 to 0.63, and for yeast, the standard is 1.20 to 1.80. Colonies resembles gram-positive organism. Hence, pipette calibrated 280 ml is used to transfer bacterial inoculum from identification tube to antimicrobial susceptibility tube. As the organism resembles gram-positive bacteria, the gram-positive identification code and the antimicrobial susceptibility test code is loaded to the appropriate tubes. The identification code contains different types of biochemical reagents in several wells. The bacterial inoculum reacts with these biochemicals and produce different colors, and thereby the bacterial species were identified. The antimicrobial susceptibility test code wells were coated with the antibiotics in different dilution, and the results of minimum inhibitory concentration is obtained using turbidometry. Now the cords were loaded into the Vitek. After loading, press the start fill button in the Vitek screen. In this loaded section, the wells in the cords were filled with the bacterial inoculum. After the signal of transfer is observed, remove the cords and transfer it to the second section. Here in this section, the tube in the cords were cut by laser beams in the machine, and then the cords were moved into the incubator. Every 15 minutes, the changes in the cords were detected by the machine. After 12 to 18 hours, the final results of bacterial species and minimum inhibitory concentration were identified. The final step of the process involves data entry of the specimen, such as date, time of collection, sample ID, patient's name, specimen source, and growth rate. The next day results, the bacterial species were identified as Enterococcus faecalis, and antimicrobial susceptibility with the minimum inhibitory concentration of the drugs were reported.